Okay, so welcome everyone to another Quantitative Methods Network talk here at the University of Melbourne. My name is Mike Zephyr. I'm an associate professor in the Faculty of Business and Economics. And today we've got a very exciting talk by a very exciting speaker. And so we've got Carsen Stringer here from the HHMI Janelia Research Campus at the Hughes Medical Institute. And she'll be talking today about Rastermap. The title of her talk is Rastermap, an embedding algorithm for interpreting high-dimensional neural activity. And Carsen's a group leader at HHMI Janelia Research Campus, and she did her postdoctoral work with Marius Pachitariu. I should have tried to pronounce that before this. And Karel, here's another one, Svoboda, is that it? Something at Janelia, and did her PhD work with Kenneth Harris at UCL. Oh, that's easier. She develops tools for understanding high-dimensional visual computations and neural representations of behavior. And you've had some really interesting, this is how I came across your work, some really interesting recent pieces in Science and Nature that provide nice critical commentaries on various kind of typical conceptualizations of neural activity and how to interpret it. And so thank you so much for doing this. I know it's kind of, it's later for you. It's past 8 o'clock. Already there in Virginia. So thank you so much, Carsen, and feel free to take it away.
Thanks, not a problem. Yeah, I'm excited to talk to a more diverse audience than I usually talk to. So I'll try again, feel free to ask me questions about the neuroscience or whenever and stop me with questions. In my lab, I'm interested in how the brain encodes the visual world and how it internally also represents the behaviors that you perform, whether you're moving or running or other things. And to understand how neurons work together to build these complex representations, we need to record many neurons. So me and others developed computational tools to allow routine recordings of tens of thousands of neurons simultaneously in the mouse brain. So I'll show you a little bit about how we do those recordings, and then I'll tell you how we analyze them, in particular an analysis that we did to find that the neural activity was in fact high dimensional in response to natural images. And then I'll tell you about a nonlinear embedding algorithm that we developed called Rastermap that exploits this high dimensional structure that we found in the neural activity that's also present in other high dimensional data sets. All right, so how do we do these recordings? We're going to use a microscope, and what we do is we make this small hole in the mouse's skull and we put a window on it. And basically the mouse lives day to day normally for months or years like this, and we can come back day after day and record from the same area over and over again. So it's a nice way to record neurons over time. And so what does one of these recordings look like? So we have this field of view here. Each of these little circles lighting up is a different neuron. And when it lights up, that means that it's firing. So it's neural activity. And so what the fields, the fields developed these great techniques to do this sort of imaging. And a lot of it was focused on kind of doing these recordings of tens or hundreds of neurons with this kind of imaging. And so what we did was we actually, instead we zoomed out and we also collected many planes simultaneously. So we reduced the amount of samples we get in time. So we only record one sample of neural activity every 300 milliseconds rather than a bit faster if we had recorded fewer neurons, but it allows us access to many thousands of neurons instead of a few hundred. And so what does one of these recordings look like? So here on the bottom, you'll see what the mouse looks like when it's hooked up to the microscope. It's free to run on this ball, air floating ball you'll see. And then each of these planes is each of these squares here. And then in the upper left, you're going to see the images that we selected from ImageNet that the mouse is seeing on a screen in front of it. So now there's no images coming in. You'll see there's still activity in cortex, but then when the images come in, bam. I mean, we're recording in visual cortex, so we expect visual responses. And we do get robust visual responses. And so the convention at the time also was so you have this many gigabytes of imaging data. And a lot of people were just circling these cells by hand, but that's not going to work when you have many thousands of neurons. So we developed a machine learning pipeline called Sweet2P, which allows you to extract these neurons from your recording quickly. and less time than it takes to do the recording. And so now you compress kind of this imaging data where instead of having these many gigabytes, you have every neuron is defined by one of these disks, which are the pixels inside that cell. And we compute the activity on every single frame that you saw when it's lighting up. That's the sum of the pixels within that cell. And so we're going to get traces now for each of those neurons, which will look like this. So every neuron is one of these traces. A subset of these neurons are shown here as these traces here. So these blips of neurons firing here. And going forward, I'm going to show these, instead of showing it as these kinds of traces, I'm going to show it as an image because it's easier to see many neurons in an image format rather than these little traces. This just kind of gives you an idea of what this activity looks like. All right, does anyone have any questions before I move on? Okay. Cool. All right. So what are we going to do with this kind of imaging data? So one of the questions we wanted to answer was kind of the first question for people who study data is like, what is the dimensionality of this data, of these responses to these natural images? And so what is its eigenvector, basically? And I'll kind of tell you why this is an important question in visual cortex in particular, the kind of scientific reason. So how is the visual, I'll tell you first how the visual system is set up to give you a little bit of background. So we have some image, like this image of a tiger that comes into your retina. It goes down your optic nerve and into your thalamus. And then from the thalamus to the visual cortex, there's kind of this 50-time expansion in the number of neurons representing the visual field then in thalamus. And so you have these many more neurons in visual cortex, and they're recurrently connected, and this is where the visual computation happens. And what's been unknown is what the purpose of this expansion is. So if this expansion, say, led to a visual cortex which had low dimensional activity, kind of the role of this expansion would be, I have low dimensional activity where 100 neurons are all representing the same thing in the visual field. Like maybe 100 neurons are always representing orange or something like that, or red or blue or other features. And that would be, this low dimensional code would mean that your neural activity would be more robust to noise. So if one of those neurons dies or there's a perturbation, the image is kind of blurry, maybe you'll have better luck reconstructing it. But there's a downside of having low dimensional activity. You can only, you can't represent as many things. If you have 100 neurons representing the same thing, you're losing the capacity of that circuit to represent a wider range of visual features. So if you have a high dimensional code, then you'll have each neuron representing different features of the visual field. And that might be better for downstream decoders in order to separate these complex image classes from each other, like to be able to tell something's a tiger or a predator or a prey. And so kind of having a high dimensional representation is what deep networks do with image data. They actually project the data into this high dimensional space. So we wanted to know, in particular, we're looking at the mouse brain, but in general, does visual cortex represent activity in a low or a high dimensional space? So we're going to determine the dimensionality of the activity looking at eigenvalues. So we have this matrix here, which is this many thousands of neurons we've recorded by visual stimuli. And we show 2,800 images in the recording. And we actually show these twice. And I won't go into kind of the way that we determine the eigenspectrum despite the fact that we have noise, but it helps to have these 2 or 3 repeats in order to better estimate the eigenvalues. But I'm happy to talk about why and how offline. But yeah, so you can pretend right now we're just going to take the principal components of the neural activity of these 2,800 images. So we want to know the dimensionality. We want to look at the principal components of the activity. So we take this activity and we're going to plot just the first 3 neurons, for instance, and each of these dots here is the response of these 3 neurons. You can think of them as the first 3 rows of this matrix, for instance. Each of these dots is a different one of these entries in each of these columns, or each of these 3 columns in these first 3 rows. So you have these different dots here, and then basically you want to take the eigenvalues of this data. So you're going to look at the principal components. And so principal component 1, for instance, is the direction that captures the most variance in the data. PC2 captures the second most, and PC3 captures the third most. And so these PCs, how long these PCs are these variances, are the variances, how much the neural activity varies along these dimensions. And these are the eigenvalues of your system. And so how these eigenvalues decay across dimensions can tell you something about the dimensionality of your system. So for instance, if you have a low dimensional system, you might only have a few eigenvalues that are positive and then the rest are 0. So it would basically be like, I have maybe 3 or 4 dimensions and those are the only ways that neurons vary, and I can reconstruct the neural activity perfectly if I know those 3 dimensions. And I wouldn't lose any information by throwing out, by basically not having these higher dimensions. Whereas if you have a high dimensional eigenspectrum, you have all of these variances are positive. And so basically each of these neurons is, you can think of as coding a different direction in stimulus space. And so if I were to throw away any of these principal component dimensions, then I would end up actually not fully having all of the information that the neural activity is representing. But at the same time, if you have this high dimensional representation, you're able to more finely represent your stimulus space. So why don't I show 2 examples of codes that are low and high dimensional, and maybe this will make a little more sense. So a low dimensional code, for instance, could be something where we have a single dimension, a stimulus dimension here. And we have each of these neurons has kind of this cosine tuning curve that varies across this one-dimensional stimulus space. And if you have these wide tuning curves, then you have this system that actually has only 2 significant dimensions here. It's a 2D system. And if we look at a random projection of this data, it looks like a circle like this, where basically if I move a little bit in stimulus space, the representation doesn't change very much. These tuning curves are wide, and I move a little bit, this neuron turns off a little bit, and then the other one turns on a little bit. And I'm just kind of moving smoothly through this space. So that's what happens in this kind of low dimensional space. And whereas in a high dimensional space, you could think of every neuron as representing representing a different stimulus along the stimulus vector. And what happens is, all right, we've defined it basically so that the variance spectrum is flat here, this eigenvalue spectrum is flat. And then if I move a little bit in this space now, one neuron turns off and another one turns on, and so on. And I basically, if I move a little bit, I'm going through this spiky ball because the representation completely changes because that neuron just completely turns off. There's no more information of where you were in the past. And if this is a continuous variable, for instance, like if I'm moving through a scene in visual space, you might not want that the representation completely changes as I move a little bit. You might think that the neuron might turn off a little bit or turn on. So this might be disadvantageous, in particular when you're trying to think about representing the visual field. So generally in the field, people have only been able to record 10 to 100 neurons, and they've mostly concluded that neural activity is low dimensional. But that's a limitation of these experiments. If you only record 100 neurons, you can only have at most 100 dimensions. If you only show 5 or 10 stimuli, you can only have 5 or 10 dimensions. So we recorded many more neurons and we showed many more stimuli than people had in the past. And so we wanted to see, okay, in this large scale data, what is the answer to this question? So does our eigenspectrum look like this, or does it look like this? And actually we found that it was neither of these things. It actually decayed as a power law, where it's it's a 1 over n power law. So you have the nth PC has, so the 10th PC has 1 over 10 variance because alpha is close to 1. So it's this 1 over n power law decay. And so this was kind of surprising to us that it was neither of these cases, and this fact that this is kind of a multi-scale eigenspectrum, that like the first 10 dimensions have as much variance as the next 100 and then the next 1,000. So you kind of have these lower dimensions that represent more of the variance in your space that change more slowly. And then you have these finer features potentially being represented in the tail end of the spectrum. And so to kind of get a better handle on this, let's make a 1D model that also has a power law decay. because it's hard to kind of think about it just in this abstract sense. So we can make a toy system where we have a 1/n power law decay, where each neuron now is a cosine tuning curve of varying frequency, and the cosine with the lowest frequency has the most variance, is this biggest one. And then the next neuron has a higher frequency and it has smaller variance, it has a smaller response, and then smaller and smaller and smaller. And these keep getting really small, and their variance is reflected along this line here. And so now we make this random projection in 3D of what these tuning curves look like as we try to move through this neural space along this one-dimensional manifold, one-dimensional stimulus dimension. Sorry. What you'll see is you mostly move smoothly, and then you've also got these little divots here that you have kind of some finer representations, but you generally are still representing this manifold in the stimulus space in kind of a smooth way, that your representation isn't completely changing as you move along this dimension. And we can make kind of a movie of this, where basically that we change alpha. So alpha is the decay of this power law. So we can think of, so if alpha is bigger, that's n to the, let's say it's n to the 4, it decays faster. So n to the 4th decays faster than n to the 3rd, which decays faster than n to the 2nd, and so on. And so we can see as we increase the amount of variance we put in our higher dimensions, which we can think of in this toy system where it's kind of like these Fourier dimensions, we're putting more and more high frequency components in, these little tiny ones that change faster and faster. What's going to happen to this curve here? And let's see, is it going to play? There we go. So now we're moving this curve up, and we're looking at how this 1D manifold changes. And so what you can see, I hope, is that it became much less smooth, and it kind of, the space is no longer representing this one-dimensional whatever stimulus feature we're looking at here. And so as you get flatter and flatter, you have more capacity, you're able to represent more different things, but at the same time you're losing this idea of kind of smoothness. So we think of neural codes as balancing capacity. You want to have as much information as possible about the stimulus space, which you can maximize by trying to decorrelate your responses as much as possible. But you also still want to have smooth representations, probably. You want to have that your representations of the world don't change drastically as some small variable changes, for instance. And that could be a variable in space. It could be a variable in image class space, like a cat. Cats can look different, and you kind of might want a smooth representation across the cat object space rather than, I mean, just to basically to do classification and these other tasks quickly, which we can do as humans. We very efficiently and quickly do these things. You want to be able to have a representation that's you probably want this smooth representation where you don't have these complicated classification boundaries that your brain has to draw for you. So yeah, does anyone have any questions? I'm going to stop here now. Has anyone ran into any power laws in their own data?
Yeah, I think in genomics, it's kind of the same. And they say look for what they call an elbow in the scree plot of variance, where it suddenly drops, and I think that's the indicative of where you start to have negligible, what they call, or noisy part of the variance in the data. So that kind of sounds familiar in that sense.
Right. Yeah. I think what surprised us is that, like, this is a log plot as well, I should specify. So it does look different probably than maybe the way you make it. But yeah, it took a long time. The elbow would kind of be around here. And so it took a long time for it kind of to get to this elbow point, which kind of surprised us. I don't know if it's the same way in genomics data.
Depends on the correlation structure in the data, obviously, and this is so what we always advise for people to look at this plot and kind of ensure that this kind of power law holds, and otherwise, there's probably less, there's less chance of having a successful low rank approximation of the data if you don't have this kind of pattern in the variance.
Cool. That's interesting. Thanks. There's a question here. Would non-smoothness mean that you are able to distinguish between different objects that look similar? So having things less smooth might mean that it would help you see things that are close to each other, but it depends on in what space things are smooth and what you're looking for. I think the problem right now is we don't really know what dimensions the smoothness is existing over. Like we think that there's kind of, we don't know for sure, but we think that to some extent there's a lower dimensional nonlinear manifold potentially underlying this neural data, but we really don't know what those dimensions are. Then I think the question becomes like, how does that influence perception? The dimensions that are represented smoothly, you might be more easily perceive them as similar rather than disparate things. And you might be able to think of psychophysics experiments where you could try to trick people depending on what nanofolds you find to be more or less smooth. But we are not there yet. There's lots of open questions in neuroscience for sure about these things. This is kind of like the first pass, and that's kind of one of the reasons we developed Rastermap, was to try to figure out kind of better what these dimensions are. Okay, so as I was saying, and I'll as well added, I don't know if that's, I'm just saying all because that's the first part of your name, sorry. Added there, you see power laws. Al. Cool, thanks. That you see power laws in lots of different data. And yeah, you'll see lots of significant dimensions, and you might see also this power law if you look at the log-log plot of your PCs, of your eigenspectrum. And so we see it in our stimulus-driven activity. So this is the result I was showing you before. We also actually see it if we turn off the lights and let the mice sit there in darkness. We also see this power law decay. And then also in zebrafish whole brain activity, so they're able to record like 60 to 80,000 neurons simultaneously. They actually also see this power law decay. It decays a bit faster, so it's more low dimensional than mouse neural activity, but it's still a power law. And then kind of the canonical example of a power law decay is the natural image spectrum, where you have the low frequency components of images have more variance than the high frequency components. And natural images also have this self-similarity property where you have these, if you look at images, for instance, of trees, you have these eigenvectors. The low D eigenvectors are going to be similar to the high D eigenvectors. As you zoom in, things are the same, look the same. So you have these kinds of properties in natural images. And then we looked at the English language. What we did was we took the correlation matrix of words that occur in the same sentence, and looked at the eigenspectrum of that correlation matrix, and that also has a power law decay. And then also if you look at nodes in a social network, you'll also see this power law decay. And so we see this is common across high dimensional data, and we want to take advantage of this structure. It gives us a clue, we don't have to build a model that works for high dimensional data where every feature is completely different. We want to take advantage of the fact that there are going to be some of these lower dimensions that have more variance, and we're going to try to create an embedding algorithm that tries to capture these slower changes across the whole population. Okay. All right. So why do we need one of these visualization algorithms? So I think anyone who's worked with high dimensional data will know it's really hard to figure out what's going on. You might be able to visualize the first PC or something, but it's going to be a lot harder to see anything past the first few PCs. So a lot of people will use nonlinear embedding algorithms like t-SNE or UMAP or these other things. And so What these algorithms do and what our algorithm is trying to do too is basically we're going to try to resort this axis here. Oh yeah, sorry, I should say this too again, because this is for the non-neuroscientists. So this is a neurons, this is called a raster plot. It's neurons by time in this case. It could be neurons by stimuli. And each of these dots is the neurons firing. And what we want to do is we want to resort this axis. We want to find an embedding, a dimension to embed the data into, in which the neurons which are correlated to each other are put close to each other in this space. And so we've created this algorithm called Rastermap, which allows neurons, which tries to embed these neurons which are correlated to each other, close to each other. And what it also tries to do is it tries to preserve these longer scale distances too. And this is something that other embedding algorithms try to do too. They try to preserve local and global distances. And this is a hard problem because you're trying to smush your data. We know the data is high dimensional and we're trying to smush it into 1 dimension or 2 dimensions or something. So it's never going to be perfect. But the goal is basically to try to preserve, as you can see, over thousands of neurons, we have neurons that are close to each other that have similar activity. And so that's kind of the goal of any of these algorithms. And there's always this trade-off in these algorithms of capturing the global structure better or the local structure better. And these kinds of things trade off in these various embedding algorithms for people who have used them. So how do we apply these kinds of global constraints? So I'll take you through how it works with this kind of toy example. Let's say we try to first cluster the data into discrete clusters using something like k-means. Then we'll get a raster plot that looks like this. We look at our correlation matrix, and it looks something like, if we do clusters like this, you're going to see these little squares along the diagonal. And then, but then in the off diagonal, if we had found a nice embedding dimension here, then the off diagonal should have been more blue. Red means positive. And so it didn't find the best dimension to embed into. And we can visualize that as well by trying to go through this dimension here through the PC space. So each of these neurons has a weight onto our first 3 principal components of the data. And so we're plotting each neuron as a point here, and we're drawing a line through these neurons depending on where they are along this embedding space. And what you can see is that as I go through this embedding space, you're kind of in this completely tangled ball here. So we haven't found the best dimension here to embed the data into to try to capture kind of the global structure of how the neural activity varies across population. And so you can start adding local constraints where you require that clusters that are close to each other are correlated to each other, basically just kind of resorting the clusters and in a way that makes them more similar, puts similar clusters close to each other, and that starts to help things a little bit. But it works even better if you start to apply these multi-scale constraints where you force these long-range clusters across long distances to be similar to each other. And then that's where you start to see these kinds of, the embedding start to separate things, like this part of the embedding is representing this part of the PC space, this part of the embedding is representing this part of the PC space, and so on. And so now I'm going to go through how we apply these multi-scale constraints. So we're going to, most embedding algorithms work on the distance matrix or the correlation matrix between points. In our case, we're talking about between neurons. In our case, we're going to reformulate it as a matrix kind of reconstruction problem, where we're trying to reconstruct our data, which is this matrix of neurons by time. And each neuron has some activity, R sub k, where this is the activity. It's going to be represented we're going to try to reconstruct it with, it's going to have a scaling term, and then it's going to have this function f, which is a function of the position in the embedding versus time. And so we're going to make f separable over time and space. Yes, that's position. So we're going to have it basically be a sum of basis functions, which are in space, and then these coefficients, which are in time. So these basis functions are going to look like this. And this might look kind of familiar if you think back to our 1D example of a power law. So we're going to have our cosine, our first basis function is going to be this cosine that changes the slowest. And then our second one is going to have 2 times the frequency and then 3 times and so on. And then we're going to restrict our coefficients. They can vary across time, but their variance has to be restricted to this power law, so that the basis function that varies the slowest has to have the most variance. So we're going to try to force neurons across a long distance to be similar to each other, because this temporal component basically is the largest at this longer scale, basically. So more neurons are influenced by this, by the first component across this embedding. And then we allow kind of these higher frequency components allow smaller groups of neurons to change together, to have their own separate activity in these higher components. And so we are going to rewrite R as a matrix, and we can rewrite B as a matrix as well, where we plug in with the X's for the neurons. And then we have A, which are our coefficients, and lambda as well. And we do this in an iterative optimization. So let's say you know A and lambda, then you can find the best B, the best X, the best position of the neuron across that embedding. And then if you know the position, you can fix the position and then recompute A. And it ends up just being a basically R times, or A transpose times R, because B is actually going to be orthonormal. All these vectors are orthonormal to each other. So you're just basically projecting back onto, oh wait, sorry, I said the wrong thing. It should be R times R transpose times B, I think, if I remember correctly. Anyway, it's simple matrix multiplications each time is the point. You're solving this least squared problem, which is simple to solve. And so that makes it fast to optimize over each iteration. And yeah, and then we also try to make it faster by discretizing the amount of points that that exist in this embedding. So we maybe only allow there to be 100 points along this embedding space. And then at the end, we'll use gradient descent to try to move these neurons to a slightly better location, rather than just being all stuck at these nodes here, to allow a continuous representation along the embedding. All right, does anyone have any questions? I'm sorry if I explained that a little confusing. We got a gene expression link.
Yeah, sorry, this is a rough plot that I just made right now, and just to show that the variance in these data sets are also, I mean, I didn't log it in here, but just to show that how we look for that elbow, which is the indication of the probably the rank of the signal part of the data.
Right, yeah, and this looks like if you took, I guess this on a log scale would look a bit more linear.
Exactly, yeah, but because this is not customary to do that, and we want people to actually see that difference, to kind of not go for too many components in the data, I guess. By default, we show it on just the regular scale.
Cool. Yeah, so I think, so I have tried running Rastermap as well, actually, on gene expression data too, and it looked okay, but I think t-SNE worked a bit better.
Yeah, and recently
that type of load. Yeah, sorry, go ahead.
Yeah, t-SNE has been a favorite up until about a year ago, and I think UMAP kind of stole the thunder, and there was there was a discussion about UMAP kind of preserving the global structure, which I think is debatable, but probably just a bit better than t-SNE at least.
Yeah, I think the default parameter. Yeah, I think UMAP has had a better publicity campaign, is what I would say recently.
Yeah.
I think that they both do very similar things.
Yeah, there was an article saying that it's just about the defaults, and starting with the same defaults, they would actually produce the same result. But I think the main argument, the way I remember when I read the UMAP paper, was that after that the manifold is being made, how that could represent the distance in the higher dimension, even with the samples or observations that are not in the vicinity of each other, kind of, the distance across the manifold representing the global distance in the higher dimension as well. But obviously, it's all debatable. I couldn't wrap my head around.
Yeah, so they have basis functions to represent their space, to do the optimization better. So maybe but they still have this problem with reconstruction, that they still don't have the exact, to reconstruct points in that space, to re-embed new points. You still, I think most techniques would use like a nearest neighbor method or something like that. I don't think they have an exact way to do that, because it's not a model of the system, I guess is what I'm trying to say. Like in this case with Rastermap, we actually have a model of what each neuron is. We have kind of a generative model of the data. I'm not saying that's better or worse, but in the case of where you're acting on the distance matrix, you're not trying to reconstruct the raw data. So you don't have a way to embed new points in that space without using I mean, not hacking necessarily, but for instance, finding nearest neighbors and putting those points near those neighbors and so on.
Yeah, I remember that was how UMAP reconstructed, using a minimum distance and then the nearest neighbors. So, I remember that in some studies, it was very sensitive to that minimum distance used, how the algorithm would reconstruct, and you could get, drastically different results based on that. So, yeah, I think they did. The way I kind of recall is that, yeah, it does use the nearest neighbors, and I don't remember them mentioning anything about trying to kind of recapitulate the generative model as well.
Yeah, and if that's not your point, then that's fine. Like if you just want to find a space where you're getting the distances recomputed, then that's fine. What helps in our case, I think the reason UMAP can perform better on neural data is trying to make this generative model, I think helps in noisy situations. So our neural data is pretty noisy, and I think it helps a bit to have this kind of this way of reconstructing things, can kind of help to smooth a little bit over that noise. Whereas if you're acting directly on the neurons, it'll be a little harder to do that. But that said, I think t-SNE and UMAP work pretty well on this data too, and I'll show you some benchmarks in a bit.
Yeah, looking forward to that.
Yeah, so I mean, it just depends. Like having this fact that we have nodes and stuff like that, we reduce our dimensionality again, and we're able to hopefully better estimate A, for instance, than maybe if we were just working on the raw neural activity. Also, it's not computationally tractable to not do it without nodes. I think there was another message. Sorry, I'm trying to open the chat. There's another question here. What informs the choice of basis functions? Yeah, okay, so the basis functions, you could choose them to be anything, but you need something that spans the space of functions across this dimension. So we want to be able to reconstruct the activity across this whole space. So the Fourier basis are an easy way to reconstruct any arbitrary function along this dimension. It could be, you could use something like wavelets or something like that, but I wouldn't expect to see huge differences in performance. I think it was stated that, oh yeah, so the A's are computed during the algorithm. So once you fix where the positions of the neurons are, then you compute A by projecting the activity into the basis function space. And then to find the positions, you basically find where the neuron is most correlated to, what position it's most correlated to, and then you put that neuron there in that correlated space with the scaling term that you find based on where you put it. Yeah, which are constrained by some initial choice. Yeah, so there are some things, there are some free parameters here that you can change in the model too, which I didn't mention. So you can actually change the decay of this. So it could be 1 over n to a different exponent than 1. So you could do 1 over n squared, And that actually changes. Yeah, unfortunately, there's no paper on Rastermap. I just saw the chat. But yeah, so basically you could change the exponent here. That's one of the parameters you can put into the model. And you can run Rastermap, by the way, it's on GitHub. It runs like any of the scikit-learn functions. It's a class like that. So you can make it n squared to try to enforce more smoothness along this axis, basically, to try to emphasize fitting global distances more than local distances. Or you could decrease it and try to fit local distances more than global distances. Okay, yeah, I think I'm kind of running out of time. Not too bad, but I just I'll show this. I don't have very many more slides. But basically, something else you can try to visualize your data and visualize this power spectrum. You can take your data and use only a few PCs to reconstruct the data. So I could take the first two PCs and use those to reconstruct the data, and then I could sort my activity along the neuron dimension and the image dimension. You can sort whatever dimension you want, basically, and then you'll get to see this activity kind of hugging the diagonal here, where basically the neural activity is slowly changing across these PCs here. But then if you take the higher PCs, you'll see that you'll start to see these clusters of activity of neurons activating to smaller subgroups of stimuli, which is kind of what we'd expect. And then you go to higher PCs, you'll see even smaller subgroups, and then even smaller subgroups if you take the higher PCs. And so this is a way to kind of visualize this, this multi-scale structure of the neural activity, that there's these low dimensions that have most of the variance that change slowly across the population, but then there's these higher dimensions that capture less of the variance and have these smaller subgroups of active neurons. And then you can also do this to sort whole brain activity. So the activity I've been showing you so far has only been in visual cortex. You can also sort neural activity recorded across the whole brain. So these are recordings that Nick Steinmetz did. And then you'll actually see neural activity. This is thousands of neurons across different brain regions that are active in similar patterns. And what we did in this paper was what we found was that if we look at the mouse's face and we look at the behaviors the mouse does, it whisks, it sniffs, it runs. We found that these behaviors we use these behaviors and we were able to predict the neural activity. And so this is what the prediction of the neural activity looks like using the behaviors alone. And we predict about 1/3 of the neural variance using the behaviors. And so across different brain areas, whether you're in visual cortex, you're in motor areas, you're in hippocampus, the memory area of the brain, all these areas are representing what the mouse is doing at any given time. And so that was an exciting result to us. It makes you rethink what you call primary sensory areas. Like visual cortex knows that the mouse is whisking. It also knows the mouse is sniffing. So what kinds of computations are actually happening in these areas that are getting direct sensory input might be very different from what we think of conventionally, where we often think a primary visual area is only computing visual information. It doesn't know anything about what you're actually doing. So this is kind of, that's why I think this result is interesting in that sense. And also kind of just generally using embeddings to visualize how well your model does, I think is a useful tool. That's part of the reason why I'm showing this. So like I've resorted this axis by Rastermap, and I can look and see kind of what subgroups of neurons I'm capturing well with this regression model using the faces and what I'm not capturing well. And it's kind of a nice way to visualize these things if you don't care about the neuroscience behind it. But yeah, it might be a useful tool. So you can also use this on whole brain zebrafish activity if you've got, so this is actually data that's freely available that you could play with if you're interested. And also all the data I've shown already is also available online. You can resort, they get 64,000 neurons recorded simultaneously. And this is what this activity looks like across time. You can see all this like, these kinds of periodic events, and that's actually different stimuli that they show to the fish. And what happens with the fish is you show it drifting gratings in different directions, and the fish will want to align to those directions or not align to those directions, depending on if it wants to swim I'm not sure if it wants to swim with the current or against the current, but basically it turns its body to try to swim in phase or out of phase with the orientations you're showing to the fish. And basically you can get, if you color the neurons along this axis with this Virdis color map here, we can basically visualize where these neurons are in the brain. And so these are 500 neurons at a time being shown here. And so you can see which subgroups of activity are localized within the brain, which ones aren't. And it's a nice way to visualize the activity. And then finally, we can run Rastermap in 2 dimensions. So we can extend by just making 2-dimensional basis functions instead of 1D basis functions. And these are basis functions taken as the principal components of natural images, but you could construct them just as sines and cosines. The algorithm actually works the same way. So this is an example here of another set of basis functions you could use. So as long as these basis functions span the possible, that they allow you to reconstruct a lot of a large space of functions, you should be fine in terms of using them, if that makes sense. Okay, so we do Rastermap on, this is now 300,000 neurons pooled over 5 different mice. These are loss of recordings that Mihaly did. And you can basically, in the GUI, you can choose clusters and you can try to look at activity and see if you can find patterns. What are these clusters responding to? And this is kind of another visualization of how high dimensional this neural activity is, that we find these different clusters, and you could even choose clusters that are right next to each other, and their activity might be very different. So there's a lot of different responses in visual cortex in response to these images that we're just starting to try to understand. And so finally, how do you benchmark these algorithms? So one way you can do it, what we did is kind of a peer prediction kind of benchmark, where you try to, you're seeing how correlated a neuron is with its nearest neighbors in the embedding. So we correlate the neuron with the average of the activity of its n nearest neighbors. And so we can start with a neuron, we can average the 1 nearest neighbor, so that will tell us, or 4 or 16, and kind of these small regions will tell us how good the embedding is working in terms of preserving local distances. And then we can start expanding that circle and see how well it's doing in terms of preserving global distances. So if we do that with the zebrafish data, you'll see that Rastermap, so they all do kind of similarly. So this is UMAP and t-SNE, and then also PCA here. So they all preserve local distances relatively similarly. And then Rastermap kind of outperforms at these medium and slightly more longer range distances here. And then if we look at this mouse data, which was even larger, this was the close to 300,000 neurons. t-SNE actually is doing better at these local distances, but then Rastermap is outperforming at these longer range distances here. And so if you want to run Rastermap on 300,000 neurons, it will work, it won't crash, and it will take around 10 minutes compared to around 70 minutes with t-SNE, or UMAP is very fast, so it'll only take 4 minutes, around the same amount of time, close to the same amount of time as PCA will take. So yeah, so it's efficient enough to run on these really large scale data sets. So that's useful if you have very large data. And so yeah, that's pretty much it. Neural activity we found is high dimensional. We've created this visualization that kind of tries to exploit this high dimensional structure that we found, that there's this power law decay. It's not just completely decorrelated and high dimensional. There's these lower dimensional components that kind of dominate the activity, or not completely dominate, but have that play a role in the activity that we're trying to capture with this power law decay in the Rastermap constraints. And so it's open source. You can download the GUI on GitHub. I mean, and it's on pip. You can pip install Rastermap. And here you can run Rastermap inside. You can load your data into the GUI and run it in there, and then you can draw clusters here. And you can also run it in a Jupyter notebook, calling it the same way you run scikit-learn functions like t-SNE or PCA. And yeah, so I'd like to thank my advisors, Marius Pakitsariou and Kenneth Harris, and also all my collaborators at UCL and at Janelia. And also I'm currently looking for postdocs and students. So if you're interested in working with this kind of large scale data and interested in developing these sorts of tools, get in touch with me. Yeah, that's it. Thanks, everybody.
Thanks so much, Carsen. That was great. Does anybody have any questions here at the conclusion of that excellent talk? Al or anybody else, do you have anything follow up before? I certainly have a question, but yeah, go ahead, Al.
Yeah, I didn't have. I mean, I just wanted to thank Carsen for the great talk, and obviously I was quite itchy to make sure that power law does hold in on a log scale. So
I'm gonna click on it now.
Put the image in there. Yeah, it's quite interesting.
Cool. Yeah,
I would be interested to see if this idea has been adopted for genomics as well. And I think it's could be a little bit tricky because there's at least in gene expression, and for now it's difficult to come up with a generative model. It's all exploratory at the moment, but we're moving towards, I think, that kind of space. So I would be interested to see if these kind of advancements and rastering would be adopted for genomics data as well. For visualization, there has been a lot of rasterization, I know that, but I'd be keen to see, if there's further developments as well. Anyways, thanks again for the great talk.
Yeah, no problem. Yeah, actually, I think that relates kind of to, someone asked this question too, like when are the coefficients sparse? And this, just like t-SNE and UMAP, it's not trying, I mean, we are using a generative model to fit it, but we're not trying to find like a sparse model underlying the data. And so if you want to do that, you might want to actually directly build a matrix factorization algorithm, something more like NMF or something more sparse than that. And that might be a better model of the underlying data, but that might not be compressible. And we probably don't expect it to be compressible below like two dimensions. Like you would need to use many more dimensions, which then actually would be less visualizable.
Exactly. Yeah, there is a high, there's an immense interest in making, I think, having a sparse representation of these components. And traditionally it was because the data were high dimensional and in the sense that there were much more features than samples. But interestingly, for example, say in gene expression space and with development of single cell gene expression profiling, so actually the old tale of I have much more features than my samples does not hold anymore, because it is now possible to profile hundreds of thousands of cells, and you would be profiling gene expression in expression for, like, say 40,000 genes or 20,000 genes. So the number of samples are growing and growing, and I'm not, probably there would be an interest, there would be less interest in making it having a sparse representation, and probably moving towards kind of being interested, as I said, in the generative process as well. Anyway, thanks again, and it was very fruitful. I'll definitely be reading up on this rasterization techniques.
Yeah, it was. Thanks, Al. It was super interesting to see that kind of, that power law, what looks like a power law emerge. I mean, obviously, there's a lot of work on allometric scaling in, like, for example, mammalian, what is it? It's like Stephen West has done a lot of research on that, right? In metabolic rates, as well as, hold on, here's a link to some of his work, which is just based on kind of a fractal theory of networks, basically. So just like the branching structure you showed in the, what were literal branches, they show it for, various kinds of, they show it in cities, they show it, these kinds of scaling laws, and like the number of gas stations and the number of freeway exits and on ramps and stuff like that. So it's interesting to see it show up in that scree plot on the log scale. One question I had, and this was the first thing that came to my mind when I read either the Science or Nature piece. I can't remember which of the two it was, and that is, I'm always kind of interested in the interplay between theory and method, and how the theories we have drive the conceptualizations that and the interpretations that different kinds of methods make available to us. So obviously for a long time, and when I took neuro classes in, like, I guess it was, the late 90s, we always heard this narrative about noise. There's a lot of noise in these neural systems. And so when you look at these patterns of activation, and obviously they were using comparatively rudimentary methods back then. And so everything looked like noise because it didn't fit the models. And their models were kind of simplistic representationalist understandings of what the brain is really doing, right? And so the brain is this kind of, it has these representational mechanisms, and boy, is it noisy. And that always struck me as being quite odd, the idea that some kind of adaptive evolutionary system would produce a noisy, wasteful, kind of computational engine. And so what you're finding, the idea that in the visual cortex, you're actually getting signals that are associated with, physical actions. I think that kind of takes us away from this theory of the brain as purely kind of a representational, a device for representing a kind of external reality. And so it's mixing signals, right? What is the animal prepared to do? What is the entity or organism prepared to do in terms of action? How is it physically situated, like the kinematics, for example, of the position of its body? And that actually is more consistent with the kind of classical pragmatist understanding of what experience is. And experience isn't just some representation of the outside world. Experience is what you get when you combine a sensory apparatus with an organism that is poised to engage in various kinds of activities. So what you see are opportunities for action, as opposed to just some abstract representation of the external world.
Right. Yeah, it definitely shapes what you see. Depending on what you're doing will change too. Like what you expect to see is something else too, that I think we think it might be a scaffold for predictive coding, that you're thinking about what's upcoming in your visual world depending on the speed that you're moving or something like that. Yeah, but I will add to that though, that this isn't all of the noise. There's still noise there.
Right.
So what people commonly thought of as noise, a good fraction of it is there are these behavioral signals, but there's still some noise left over that we still don't know what it is. And to some extent, it might just be single neuron noise. And it's just a consequence of these are imperfect. They're not perfect firing neurons, but we have a lot of neurons. So it's not the end of the world either. That we have enough neurons representing each of these spaces that if one of them isn't firing perfectly, it's not a big deal. And if we have smooth representations, it's also not a big deal then too.
Right. And it's only a partial representation of the brain.
Yes.
So there's that.
Yeah, that too. Like what is noise might be from other brain areas telling us other things. And it could be, gut-brain interaction is fascinating. And that's another thing too. Like there's thirst signals in visual cortex too that people have studied. And that's not something we can get from the mouse's face, or we don't think we can.
That's right.
I highly doubt we can.
That's right.
So yeah, I don't personally work on gut-brain interactions, but it is a really exciting topic. Unfortunately, it's a lot deeper in the brain, so it's a lot harder to record from. And so a lot of these techniques are good for cortical areas. So that's why studying visual cortex is pretty easy with these kinds of microscopes.
Yeah, it's really cool. Okay, anybody else? I don't want to keep it for too long. I realize it's like 9:03 where you are. PM. Does anybody else have any questions before we let Carsen kind of retire into her evening? No? Okay. Well, thank you so much. This has been, such a great pleasure, and obviously
nice meeting everyone.
So we'll, yeah, that's right. Okay, I'll stop this.